presentation is from Dr. Cooke. He's the leader of our center, and he's going to give us a very important update on the work that is undergoing in our center. Thank you, Dr. Cooke.
Thank you, Johnny. For those of you who haven't joined us before, I'm John Cooke. I'm chair of the Department of Cardiovascular Sciences, and you are here at the Center for Cardiovascular Regeneration at our weekly scientific session. Today I'm going to start by introducing this concept of S-nitrosylation in reperfusion and regeneration as part of our journal club that we are having today, and also as an introduction to the work that Dr. Shu Meng is going to present a little bit later. So the overview for my talk is that we will be addressing S-nitrosylation and epigenetic plasticity, something that you're familiar with. S-nitrosylation and perfusion, which you may not be, it's a subject of the journal club today, and the potential clinical implications. So the paper that I chose for our journal club today is this one from Jonathan Stamler's group at Case. Stamler has been doing a lot of work for decades in S-nitrosylation and its effect on protein activity. This paper is a continuation of his work on hemoglobin S-nitrosylation, in this case its role in cardioprotection. So why this article for a journal club? Well, as you know, we have 3 criteria for our journal club. One, it should be from a high-impact journal. So this one's from Journal of Clinical Investigation, which has an impact factor of 12.6. That's pretty good. It should be a recent paper. This one is recent, from December 2016. And it should be relevant to our work. So how is this paper relevant to our work? Well, as you're going to be hearing from Dr. Shu Meng momentarily, nitrosylation plays a critical role in transdifferentiation, in the nuclear reprogramming that occurs for the transdifferentiation of one somatic cell into another. And specifically, Shu is working on the effects of innate immune activation and endothelial cell transdifferentiation factors for this process of transdifferentiation of fibroblasts to endothelial cells. Her project springs from a larger program at our center looking at the role of innate immunity in nuclear reprogramming. And as you know, we've termed this process by which innate immunity induces epigenetic plasticity. We call this transflammation. So this is a process we first described in a Cell paper in 2012. We find that pattern recognition receptors on the cell can respond, of course, to pathogen-associated molecular patterns and damage-associated molecular patterns, triggering a cascade of signaling that's well known, well described to cause the activation of NF-kappa B, IRF3, transcriptional activators that induce the immune response, the release of inflammatory cytokines. But what we found, which was not known, is that this same process causes global changes in epigenetic modifiers that induces epigenetic plasticity, so induces an open chromatin configuration. And this process is absolutely critical for the cell plasticity that's needed for induction of pluripotency or for transdifferentiation. So today we're going to be focusing on nitrosylation and transdifferentiation. And the transdifferentiation that we're interested in is that which produces induced endothelial cells. So Shu has a nice protocol for generating induced endothelial cells, which involves activation of innate immunity, and then the addition of factors that are basically cues for transdifferentiation of the fibroblast endothelial cells. And the endothelial cells that she derives are very similar to genuine endothelial cells. So in this hierarchical clustering of the RNA-seq data, you can see that the induced endothelial cells in the 2 middle panels are very similar to human microvascular endothelial cells and very different from the parental fibroblasts here. And histologically, these induced endothelial cells resemble genuine endothelial cells. They express all the immunohistochemical markers, CD31. They take up acetylated LDL. They form tubes in matrigel. They make nitric oxide, just like normal endothelial cells, and very different from fibroblasts, which do not. Shu is helped by Iris in using the hind limb ischemia model. Shu has shown that these cells, induced endothelial cells, can actually improve perfusion. So here's mean perfusion over time after ligation of the femoral artery. That causes a 80% reduction in blood flow. Over time, that improves. Control is in black. Green is the injection of induced endothelial cells into the ischemic hind limb. Red is the injection of genuine endothelial cells into the ischemic hind limb. And you can see there's some benefit of the injection of the induced endothelial cells and human microvascular endothelial cells with an increase in capillary density and a reduction in the damage as shown by the hind limb ischemia score. Now, Shu's recently published this work on the role of iNOS in this process. So she's shown that iNOS increases during the transdifferentiation process. eNOS, of course, is expressed by endothelial cells, but eNOS doesn't really come into play until very late when the transdifferentiation process is terminated in the generation of an endothelial cell. Neuronal NOS is not present. So iNOS is activated early, and Shu was interested to know if that was important for the transdifferentiation, and indeed it is. This is work with iNOS knockout fibroblasts, and in these, in the normal fibroblasts, the transdifferentiation results in a yield of 4.5% IECs after 4 weeks. It's not a lot, but recently she's improved the transdifferentiation protocol so she can get yields up to 10 to 15%. Nevertheless, it's with iNOS knockout, she gets far less induced endothelial cells. In fact, iNOS knockout almost abrogates transdifferentiation. Now, one thing that's really interesting about this is that it appears that iNOS translocates to the nucleus during this process. So this is before activation of innate immunity. This is after activation of innate immunity, and you can see that that iNOS is translocated to the nucleus with stimulation of innate immunity. So stimulation of innate immunity induces this translocation of iNOS to the nucleus, and the question that Shu had was how might this process, how might iNOS translocation enhance epigenetic plasticity? Well, here we are again with this diagram. Pattern recognition receptors activate NF-kappa B, IRF3 and induce iNOS. Now, how can iNOS cause changes in epigenetic plasticity that will permit this transdifferentiation to occur? Well, it could be mediated by cyclic GMP. NO stimulates guanylate cyclase to produce cyclic GMP. It could be through S-nitrosylation. It could be through nitrotyrosinylation, or some physical association of iNOS with the epigenetic modifiers potentially that would result in this epigenetic plasticity. But to make a long story short, so we can move on to the journal club, what Shu has found is that this polycomb complex, which includes this ring 1A protein that holds the polycomb complex together, is markedly affected by nitrosylation, by iNOS. So this complex is important for suppressing chromatin. It induces an H3K119 ubiquination. So polycomb complex has this effect, H2A K119 ubiquination, which stabilizes this suppressive mark. This is a suppressive mark laid down by the PRC2 complex, H3K27 trimethylation. So this complex is very important for stabilizing this suppressive mark. And what she has shown is that the effect of nitrosylation of ring 1, first of all, iNOS associates with ring 1, nitrosylates it, and it inhibits this process of H2AK119 ubiquination. So this stabilization of the suppressive mark is not maintained, and this suppressive mark is lost, and allowing the chromatin to assume a more open configuration. So basically what Shu has added to our story is that by activating innate immunity, iNOS binds to and nitrosylates ring 1, and that basically destabilizes polycomb complex 1, reduces the suppressive marks, and allows for epigenetic plasticity. Basically opens up the chromatin. So iNOS nitrosylation of epigenetic modifiers may play a very important role in opening up the chromatin. So now, you know, with this knowledge, we can directly reprogram fibroblast endothelial cells, and that may have important utility for fibrotic diseases, for restoring normal tissue regeneration. And Johnny, of course, and Polish are working on the role of iNOS in tissue regeneration. We're not going to be hearing about that today. That's early days yet, but they have found, interestingly enough, that in cell culture and in animals, there seems to be an optimal zone for activation of innate immunity and this epigenetic plasticity. Well, let's get to the journal club. And this is Jonathan Stamler pictured here. He's the senior author on this paper. He's made spectacular contributions to our understanding of the role of NO synthases on protein modifications. So S-nitrosylation is a protein modification similar in many respects to phosphorylation. So you know phosphorylation changes the activity of kinases. There are also, just as there are dephosphorylases, there are denitrosylases. So the nitrosylation of proteins is a reversible process that modifies their activity. So he's primarily studied these signaling cascades in the cytoplasm. That's where most of his work has been. We're moving into the nucleus. We're going to see what role this system has in the nucleus and in modification of epigenetic factors. Dr. Stamler has also made an amazing contribution to our understanding of oxygen transport. It turns out that in addition to binding oxygen, hemoglobin binds NO. And it plays a very important role in oxygen transport. So let me just take you through this diagram, which summarizes the work that he's been doing in the past 15 years on the effects of NO on hemoglobin. In the oxygenated vessel, for example, in the lung, where there's high concentrations of oxygen, hemoglobin is in this R state, and it has high affinity for oxygen, so it's binding oxygen. But it also has high affinity for nitric oxide, and it binds NO, the heme group. The heme moiety binds NO, so you have a nitrosoheme. And then as the blood circulates through the body, there's an intramolecular transfer from the heme group to this cysteine group, cis 93, on the beta globin chain. And so that intramolecular transfer then, by the time the blood reaches a deoxygenated part of the circulation, you have a situation where in this state the hemoglobin assumes a different configuration. It loses its affinity, or it has reduced affinity for oxygen, And so basically it delivers oxygen to an area where there's poor oxygen or desaturation. And you also have release of nitric oxide into the vessel wall. Now what the result of that is that in this area of reduced oxygen, you have release of oxygen. You also have release of NO to induce a vasodilation. So where there is a lot of oxygen, the blood cells, the hemoglobin has great affinity for oxygen. It also has great affinity for nitric oxide. There's basically a loss of NO from the blood vessel and some vasoconstriction, whereas in an area where you have poor oxygenation, you have release of oxygen and you have release of NO. So you have oxygenation and you have vasodilation. Now, in previous studies, he's developed a mutant mouse where the cysteine is mutated to alanine on the beta-globin chain. So this Cis93 is mutated to alanine. And he's previously shown that abrogates the NO carrying capacity of the hemoglobin. And so it abrogates hemoglobin nitrosylation. And the question in this paper was, could this, in this mutant mouse, what would be the response to ischemia, to myocardial ischemia? So that was the question. So they used this ischemia perfusion injury model in the mouse heart. And in this model, the mouse is anesthetized, chest is opened up, the left anterior descending artery is ligated, and in some cases, they put in a little tube next to the ligation, right next to the vessel, so that that tube can be pulled out and the reperfusion can occur. So it's a model of ischemia reperfusion. Now, the first thing that they observed in this mouse model was that by comparison to the control animals, and somewhat surprisingly, these animals with the mutation, where the alanine was, or rather the cysteine was mutated to alanine, had more collaterals. So the collateral channels, this is the LAD, and it's ligated here. There's more collateral channels forming in these mutant animals. And they thought, well, perhaps this is a form of compensation for basal myocardial hypoxia. These animals are not very good at vasodilating through this nitrosyl hemoglobin mechanism. So maybe they develop more collaterals. In any event, this is the data showing greater collaterals. They had two types of mutants. It doesn't really matter. When you see 93A, that's the mutation of the cysteine to alanine, and it doesn't really matter. These two groups are pretty much the same physiologically. Despite more collaterals, there was increased mortality in the mutant mice. So this is the mortality from the ischemia reperfusion, and you can see that there's a significant increase in mortality in the mutant mice. Now, to understand how much damage was being done with the ischemia, they used a classic methodology. What they did was to, after the ligation and after the ischemia reperfusion, when they harvested the tissues, they once again ligated the previously ligated vessel and then perfused trypan blue. And you can see that in this case, the trypan blue will perfuse the open coronary artery. It won't perfuse the ligated coronary artery. So you can see what part of the heart was getting perfused and what was not getting perfused. And then they can do a TTC stain to see how much of the myocardium was viable. That stain stains the viable myocardium red, and the pale myocardium is necrotic. So this is the blue area in these hearts are the area that was perfused. The non-blue area is the area at risk. So that was the whole area at risk of the myocardium after the ligation. The white part is the necrotic area, and the red part of this stain shows the viability of the myocardium in red. And you can see at a glance that this wild type had a similar area at risk. The area at risk is the non-blue area, that is area at risk. So they had the same area at risk, so the experimental ligation caused a similar risk to the myocardium. But the damage in the mutant mice is greater. You can see there's more pale area here than in the control animal. That is shown in F, panel F on this slide. This shows the area of necrosis, for example, in the wild type in red and in the mutant in green. So this ratio of area of necrosis over the entire left ventricle is increased in the mutant mice. So they had more damage. And that was in the animals that did not have collaterals. I failed to mention to you that some animals, there were some animals without collateral circulation, others that had developed collaterals. I previously mentioned that the mutants had more collaterals. So that had some modifying, mitigating effect. In the mutants that had some collaterals, there was somewhat less damage. But even still, they had more damage than the wild-type mice. So basically what this data shows is that the mutant mice have more damage, more necrosis with the same amount of ischemia. They also looked at function, and in panel G, you can see in red the control animals after this ischemia reperfusion injury, and in green the animals that had the mutant animals. And you can see that the mutant animals have a markedly reduced ejection fraction by comparison to the control animals. They have a markedly reduced fractional shortening. That's also a measure of contractility. Whether they had collaterals or not, the mutant mice did worse. And then LV wall thickening, again, it's a measure of the function of the myocardium. Its ability to contract is also impaired in the mutant mice, as shown in green. So to conclude, they basically showed that with the same amount of ischemia, there was more damage and there was reduced function in the animals with this hemoglobin mutation. So they looked at another model in the mouse of heart failure. This is acute pressure overload. And in this model, one induces a constriction of the aorta. It's called a transverse aortic constriction model, or TAC model. And that causes an acute pressure overload in the heart that can acutely cause heart failure and chronically cause left ventricular hypertrophy, pericardial effusion, lung edema, and mortality. So what happens to the mutants when they perform this procedure? So at 2 days after the procedure, by echocardiography, they were able to document a reduction in ejection fraction that was more severe in the mutant mice with the mutant hemoglobin. There was a reduction in fractional shortening, again a measure of contractility, and a reduction in cardiac output. Furthermore, there were other changes that suggested adverse remodeling. So this is the end systolic volume of the left ventricular chamber. So an increase is not good. That means your heart is dilating, and it indicates failure. And then they also measured the heart weight over the body weight. That's a measure of left ventricular hypertrophy, and that increases in the mutant mice. And they also measured lung weight over body weight. So lung weight, when it increases, in this model is due to edema. Now let me show you that actually. You can actually see it grossly, the edema. This is open chest of the mouse, and you can see the edema in the lung. It shouldn't look, this is basically what a congested lung looks like. Mortality was increased in the mutant mice. Again, 93A, the important, these are two different forms of the mutant hemoglobin, but in each case the cysteine is replaced by an alanine. So both of the green and blue are mutant animals, mutant hemoglobin. So survival over time was pretty good in the wild type animals, but in the mutant animals there was significant mortality over time, as shown in this kind of Kaplan-Meier analysis. Ejection fraction I've already shown, it was reduced. It's still reduced at 30 days, and fractional shortening is reduced. Cardiac output's reduced in the mutants, and diastolic and systolic volume is increased, and heart weight's increased, and lung weight's increased. All measures of heart failure. So to conclude, S-nitrosylation of epigenetic modifiers is important for nuclear reprogramming and phenotypic fluidity. That's the work of Shu and Johnny and Polish, and really is a very nice story. S-nitrosylation plays a role in myocardial perfusion, and both of these processes may play an important role in the recovery from ischemic injuries. So when we think of an individual that's in the midst of a myocardial infarction, we need to restore perfusion, and we also need to help the heart regenerate. And the knowledge that we gained from the studies of Stamler and the work that we're doing are potentially could lead to new therapeutic avenues for myocardial infarction. So with that, I will end, and we'll take a few questions before we go on to Shu's talk. Thank you.
Many thanks. So I have one question. So did the author try to, they have any idea how to improve reperfusion or the recovery from after injury by increasing iNOS activity or NO presence on the hemoglobin to try to improve recovery after injury? Mm-hmm. Because they show that when they knock down, let's say they knock down this capability of NO to bind to hemoglobin, there is this failure of the heart to recover after injury.
Yeah.
Did they try the other side? So did they try to improve after injury?
It's a good question. I mean, they just hold out. They didn't actually specify what could be done, but it's in this model, it's probably eNOS, endothelial NO synthase, that's playing the most important role, because it's release of NO from the endothelium that the red blood cells are taking up. By the way, it's quite interesting that this cysteine 93 that is nitrosylated, that is conserved throughout mammals and birds, that particular cysteine. And it's really the only residue that's conserved on hemoglobin, except for those residues that are important for oxygen carrying, for the heme interaction. This is highly conserved. So that indicates its evolutionary importance. I think Dr. Bruno had a question.
Yeah, I have a couple questions. When they looked at that side, well, first did they look at the phenotype of the animal, and when they looked at the cysteine to alanine?
They actually, that's a great question. In previous papers, they've characterized this mouse a little bit more, and it does have some basal alterations. And I showed you one. It has this increase in collaterals. It's thought to be maybe a response to the baseline ischemia.
So this hemoglobin, in addition to oxygen, carries iron. Did they see if that function is also altered in terms of carrying or releasing iron?
I don't think that's altered, but I do believe they've shown that oxygen release, oxygen transport is impaired.
And so they see necrosis even though the level of ischemia is the same. Did they look mechanistically why?
Presumably it's due to reduced oxygenation, reduced release of oxygen, and reduced vasodilation.
Because they did not look specifically at level of oxygen or red blood cells
Yeah.
or the amount of
I
oxygen?
believe they've done that
They've
in
done that.
past studies, characterized the release of oxygen, characterized oxygen transport, characterized the vasodilation that occurs in these animals with hypoxia. So the hypoxic vasodilatation, I do believe they've shown that's impaired in these
And the
animals.
body has ways of bouncing back. For instance, if you can carry a lot of hemoglobin, red blood cells tend to have a larger corpuscular body, or they might increase in numbers. Or so did they see something like that, some sort of compensatory mechanism?
I don't know. That's a good question. Whether it was some change in the red blood cells themselves, I don't know.
And the heart was mainly influenced. Were there any other organs that were?
Not in this paper. They didn't look at that. Yeah, though all of those questions, I think, they've addressed in previous publications, but not specifically in this paper.
Is this a mutation that occurs naturally? I mean, is there any disease associated with this, or is this something that?
Yeah, I don't know. That would be interesting to know if
It's
there's.
very interesting.
Yeah.
Another question? Okay, we can go forward with the second presentation today.
I think we'll close this out. Shu has got some confidential information that we'll just share amongst our group. So I think we'll thank Tyree for the videotaping and
Tell goodbye.
what's that?
Tell goodbye.
Okay. Thank you for joining us. This concludes our presentation today from our Center of Cardiovascular Regeneration. Thank you.